Godzilla is back and he's bigger than ever. The evolutionary biology of the monster Godzilla first made his debut in 1954. At inception, he was a 50-meter-tall metaphor for indiscriminate destruction, particularly U.S. hydrogen bomb testing in the Marshall Islands, which, in the film, destroyed Godzilla's deep-sea ecosystem. 65 years and 35 films later, Godzilla is back and bigger than ever in Godzilla, King of the Monsters. At 119.8 meters tall, Godzilla battles it out for supremacy against three god-sized monsters, all with the future of humanity at stake. Film critics and fans have long observed that Godzilla has been getting larger over time, as buildings become taller. In fact, Godzilla has evolved 30 times faster than other organisms on Earth, according to a team of Dartmouth scientists whose findings are published in Science. The researchers propose that Godzilla has been evolving in response to a spike in humanity's collective anxiety. They used U.S. military spending as a proxy for our collective anxiety and found a strong correlation between it and Godzilla's body size between 1954 and 2019. If Godzilla is the embodiment of our anxiety, they argued, then our collective anxiety appears to be spiking as it did during the nuclear age of the 1950s. If one accepts that Godzilla is a ceratosaurid dinosaur from the Jurassic period, as argued in the film series, then he represents a sensational example of evolutionary stability over a span of at least 145 million years. Yet Godzilla has doubled in size since 1954, far surpassing the rate of evolution observed in 2,500 natural organisms today. Godzilla's body was consistent for some 150 million years until 1954, suggesting a sudden and strong selective pressure on body size during the past 65 years, says co-author Nathaniel J. Dominey, the Charles Hanson Professor of Anthropology and a professor of the Ecology, Evolution, Ecosystems and Society graduate program at Dartmouth. Dominey co-authored the study with Ryan Kalsbeek, an associate professor of biological sciences and of the Ecology, Evolution, Ecosystems and Society graduate program at Dartmouth. The co-authors add that Godzilla endures as a cultural icon because it is a fable with a lesson for our times. How old are your organs? To scientists' surprise, organs are a mix of young and old cells. Scientists once thought that neurons, or possibly heart cells, were the oldest cells in the body. Now, Salk Institute researchers have discovered that the mouse brain, liver and pancreas contain populations of cells and proteins with extremely long lifespans, some as old as neurons. The findings, demonstrating age mosaicism, were published in Cell Metabolism on June 6, 2019. The team's methods could be applied to nearly any tissue in the body to provide valuable information about lifelong function of non-dividing cells and how cells lose control over the quality and integrity of proteins and important cell structures during aging. We were quite surprised to find cellular structures that are essentially as old as the organism they reside in, says Salk Vice President, Chief Science Officer Martin Hetzer, senior author and professor. This suggests even greater cellular complexity than we previously imagined and has intriguing implications for how we think about the aging of organs, such as the brain, heart and pancreas. Most neurons in the brain do not divide during adulthood and thus experience a long lifespan and age-related decline. Yet, largely due to technical limitations, the lifespan of cells outside of the brain was difficult to determine. Biologists have asked, how old are cells in an organism? There is this general idea that neurons are old, while other cells in the body are relatively young and regenerate throughout the organism's lifetime, says Rafael Araujo e Drigo, first author and Salk staff scientist. We set out to see if it was possible that certain organs also have cells that were as long-lived as neurons in the brain. Since the researchers knew that most neurons are not replaced during the lifespan, they used them as an age baseline to compare other non-dividing cells. The team combined electron isotope labeling with a hybrid imaging method, MIMS-M, to visualize and quantify cell and protein age and turnover in the brain, pancreas and liver in young and old rodent models. To validate their method, the scientists first determined the age of the neurons, and found that, as suspected, they were as old as the organism. Yet, surprisingly, the cells that line blood vessels, called endothelial cells, were also as old as neurons. This means that some non-neuronal cells do not replicate or replace themselves throughout the lifespan. The pancreas, an organ responsible for maintaining blood sugar levels and secreting digestive enzymes, also showed cells of varying ages. A small portion of the pancreas, known as the islets of Langerhans, appeared to the researchers as a puzzle of interconnected young and old cells. Some beta cells, which release insulin, replicated throughout the lifetime and were relatively young, while some did not divide and were long-lived, similar to neurons. Yet another type of cell, called delta cells, did not divide at all. The pancreas was a striking example of age mosaicism, i.e., a population of identical cells that are distinguished by their lifespans. Prior studies have suggested that the liver has the capacity to regenerate during adulthood, so the researchers selected this organ expecting to observe relatively young liver cells. To their surprise, the vast majority of liver cells in healthy adult mice were found to be as old as the animal, while cells that line blood vessels, and stellate-like cells, another liver cell type, were much shorter-lived. Thus, unexpectedly, the liver also demonstrated age mosaicism, which points to potential new paths of regenerative research for this organ. On a molecular scale, a selection of the observed long-lived cells contained protein complexes displaying age mosaicism. For example, the primary cilia, hair-like appendages on the outside of cells, of beta cells in the pancreas and neurons contained protein regions of vastly different lifespans. In stark contrast, the cells in the liver contained no long-lived proteins at all. Thanks to new visualizing technologies, we are able to pinpoint the age of cells and their supramolecular complexes more accurately than ever before. This opens new doors for studying all cells, tissues and organs in normal and in disease states, says Mark Ellisman, Distinguished Professor of Neurosciences at UC San Diego's School of Medicine and co-leader of the study with Hetzer. His laboratory, the National Center for Microscopy and Imaging Research, developed and provided the new tissue imaging methods for correlated multi-scale and multimodal microscopy. These methods provided the key new and enabling technologies that allowed this study to be carried out. Determining the age of cells and subcellular structures in adult organisms will provide new insights into cell maintenance and repair mechanisms and the impact of cumulative changes during adulthood on health and development of disease, adds Hetzer. The ultimate goal is to utilize these mechanisms to prevent or delay age-related decline of organs with limited cell renewal. Next, the authors plan to decipher the difference in lifespans for nucleic acids and lipids. They also want to understand how age mosaicism relates to health and diseases such as type 2 diabetes. Other authors included Swati Tyagi of Salk, Varda Lev Ram, Ranjan Ramachandra, Thomas Deerink, Eric Bushong and Sebastian Fan of UC San Diego, Victoria Orphan of the California Institute of Technology, and Claude Lechain of Brigham and Women's Hospital. New evidence links ultra-processed foods with a range of health risks. Two large European studies published by the BMJ today find positive associations between consumption of highly processed, ultra-processed foods and risk of cardiovascular disease and death. The researchers say further work is needed to better understand these effects, and a direct causal link remains to be established, but they call for policies that promote consumption of fresh or minimally processed foods over highly processed foods. Ultra-processed foods include packaged baked goods and snacks, fizzy drinks, sugary cereals, ready meals containing food additives, dehydrated vegetable soups, and reconstituted meat and fish products, often containing high levels of added sugar, fat, and/or salt, but lacking in vitamins and fiber. They are thought to account for around 25 to 60% of daily energy intake in many countries. Previous studies have linked ultra-processed foods to higher risks of obesity, high blood pressure, high cholesterol, and some cancers, but firm evidence is still scarce. In the first study, researchers based in France and Brazil assessed potential associations between ultra-processed foods and risk of cardiovascular and cerebrovascular disease, conditions affecting blood supply to the heart and brain. Their findings are based on 105,159 French adults, 21% men, 79% women, with an average age of 43 years who completed an average of 6 24-hour dietary questionnaires to measure usual intake of 3,300 different food items, as part of the Nutrinet Santé study. Foods were grouped according to degree of processing and rates of disease were measured over a maximum follow-up of 10 years, 2009 to 2018. Results showed that an absolute 10% increase in the proportion of ultra-processed food in the diet was associated with significantly higher rates of overall cardiovascular disease, coronary heart disease, and cerebrovascular disease, increase of 12%, 13%, and 11% respectively. In contrast, the researchers found a significant association between unprocessed or minimally processed foods and lower risks of all reported diseases. In the second study, researchers based in Spain evaluated possible associations between ultra-processed food intake and risk of death from any cause, all-cause mortality. Their findings are based on 19,899 Spanish university graduates, 7,786 men, 12,113 women, with an average age of 38 years who completed a 136-item dietary questionnaire as part of the Seguimiento Universidad de Navarra (SUN) study. Again, foods were grouped according to degree of processing and deaths were measured over an average of 10 years. Results showed that higher consumption of ultra-processed foods, more than 4 servings per day, was associated with a 62% increased risk of all-cause mortality compared with lower consumption, less than 2 servings per day. For each additional daily serving of ultra-processed food, mortality risk relatively increased by 18%, a dose-response effect. Both studies are observational, so can't establish causality, and there's a possibility that some of the observed risks may be due to unmeasured confounding factors. Nevertheless, both studies took account of well-known lifestyle risk factors and markers of dietary quality, and the findings back up other research linking highly processed food with poor health. As such, both research teams say policies that limit the proportion of ultra-processed foods in the diet and promote consumption of unprocessed or minimally processed foods are needed to improve global public health. This view is supported by Australian researchers in a linked editorial, who say the dietary advice is relatively straightforward: eat less ultra-processed food and more unprocessed or minimally processed food. They say future research should explore associations between ultra-processed food and health harms in different populations around the world, and examine how harm occurs, for example by changing the gut microbiome in ways that could disturb energy balance. In the meantime, policymakers should shift their priorities away from food reformulation, which risks positioning ultra-processed food as a solution to dietary problems, towards a greater emphasis on promoting the availability, affordability, and accessibility of unprocessed or minimally processed foods, they conclude. Is there a limit to human endurance? Science says yes. From the Ironman triathlon to the Tour de France, some competitions test the limits of even the toughest endurance athletes. Now, a new study of energy expenditure during some of the world's longest, most grueling sporting events suggests that no matter what the activity, everyone hits the same metabolic limit, a maximum possible level of exertion that humans can sustain in the long term. When it comes to physical activities lasting days, weeks and months, the researchers found, humans can only burn calories at 2.5 times their resting metabolic rate. Not even the world's fastest ultramarathoners manage to surpass that limit, the researchers found. This defines the realm of what's possible for humans, said study co-author Herman Poncer, an associate professor of evolutionary anthropology at Duke University. Beyond the threshold of 2.5 times a person's resting metabolic rate, researchers found, the body starts to break down its own tissues to make up for the caloric deficit. One explanation for this limit may be the digestive tract's ability to break down food, said team leaders Poncer and John Speakman of Scotland's University of Aberdeen and the Chinese Academy of Sciences. In other words, eating more won't necessarily help someone make Iditarod history. There's just a limit to how many calories our guts can effectively absorb per day, Poncer said. The results will appear online June 5 in the journal Science Advances. For the study, the team measured daily calories burned by a group of athletes who ran 6 marathons a week for 5 months as part of the 2015 race across the USA, a 3,000-mile race from California to Washington, D.C. The team also considered other feats of human endurance, including punishing 100-mile trail races and pregnancy. When they plotted the data over time, they found an L-shaped curve. The athletes' energy expenditure started out relatively high but inevitably plunged and flattened out at 2.5 times their basal metabolic rate for the remainder of the event. Co-author Caitlin Thurber analyzed urine samples collected during the first and final legs of race across the USA. After 20 weeks of running back-to-back marathons, the athletes were burning 600 fewer calories a day than expected based on their mileage. The findings suggest that the body can downshift its metabolism to help stay within sustainable levels. It's a great example of constrained energy expenditure, where the body is limited in its ability to maintain extremely high levels of energy expenditure for an extended period of time, Thurber said. You can sprint for 100 meters, but you can jog for miles, right? That's also true here, Ponser said. All the endurance events followed the same L-shaped curve, whether the athletes were hauling 500-pound sleds across Antarctica for days in sub-freezing temperatures, or cycling the Tour de France in summer. That finding challenges the idea, proposed by previous researchers, that human endurance is linked to the ability to regulate body temperature. One limiting factor for endurance events, researchers found, lies in the digestive process, the body's ability to process food and absorb calories and nutrients to fuel bodily processes. Interestingly, the maximum sustainable energy expenditure found among endurance athletes was only slightly higher than the metabolic rates women sustain during pregnancy. This suggests that the same physiological limits that keep, say, Ironman triathletes from shattering speed records may also constrain other aspects of life too, such as how big babies can grow in the womb. As far as the researchers know, no one's ever sustained levels beyond this limit. So I guess it's a challenge to elite endurance athletes, Ponser said. Science works when you're proven wrong. Maybe someone will break through that ceiling someday and show us what we're missing.